In today's video, we'll learn how to combine DNA sequences into one larger sequence. Imagine you have performed DNA sequencing. Then instead of receiving one long DNA sequence, you'll receive it in smaller pieces. This happens quite often in bioinformatics, and our challenge is simple. How do we combine these fragments into one continuous DNA sequence? Let's think before writing any code. Suppose the first fragment is ATGC and the second fragment is CGTA. If these belong together, what should the final DNA sequence look like? Try answering before Python does. Visually, we are simply placing one DNA fragment immediately after the other. Nothing is changing inside either fragment. We are not editing them or rearranging them. We are only connecting them. Now let's recall something from previous lessons. A DNA sequence is just text as far as Python is concerned. And Python stores text using the string data type. So if DNA sequences are strings, maybe Python already knows how to join strings together. Here we are simply creating two variables. One stores the first DNA fragment and the other stores the second DNA fragment. Nothing new is happening yet. We are just preparing the pieces. Now comes the interesting part. If Python knows these are strings, how do we tell it to connect them? And can you guess which operator we should use? We use the plus sign between the two string variables. And then we store the result in a new variable called full sequence. Finally, we ask Python to display the completed DNA sequence. And Python prints ATGCCGTA. Notice that both fragments appear together exactly in the same order. The plus sign might look familiar because you have seen it in mathematics. But Python doesn't treat it always as addition. In our case, since both the values are strings, so Python understands that we want to join them together and instead of adding numbers. In Python, the meaning of an operator depends on the type of data it's working with. Now let's see if you have understood the idea or not. Before running the next cell, try predicting the answer if we join GGA and TTC. What sequence should Python produce? Now let's ask Python and compare its answer with your prediction. If your prediction was GGA TTC, excellent. That means you have understood what string concatenation does. Now notice what's visually happening. Python takes the first string exactly as it is, then immediately places the second string after it. And it doesn't insert any spaces, commas, or any extra characters. It simply joins them together. This idea of concatenation appears throughout bioinformatics. Many analyses involve working with DNA fragments. While real genome assembly is much more sophisticated than simply joining strings, today's lesson introduces the fundamental idea of building longer DNA sequences from smaller pieces. Now let's summarize. DNA sequences are strings. The plus sign joins two strings together. And this process has a special name, string concatenation. This is one of the simplest but most frequently used operation when working with biological sequence data.